Hello everyone, my name is Mr. Ron and I will be giving you guys a lecture today over biomacromolecules, their properties, structure, and function. So the first biomolecule we're going to talk about is carbohydrate. And carbohydrates include sugars and polymers. These guys are comprised of carbon, hydrogen, and oxygen in a specific format. So we've got monosaccharides, who are simple sugars. If we break down that word, mono means one, saccharide means sugar, so we have one sugar. The molecular formula is with multiples of carbon, hydrogen, and oxygen. So if I have 2 carbon, I'm going to have 4 hydrogen and 2 oxygen. It is pretty standard. The most common monosaccharide is going to be glucose. And this serves as a nutrients and fuel for almost all cells. We see this glucose used in cellular respiration, which is what cells do to produce ATP, or the energy source, to keep them going. This can also serve as a building block for amino acids, or as monomers for di and polysaccharides. So let's break down those words again. Mono means one, monomers means one block or one thing. For di means 2, and poly means multiple, so 2 or multiple sugars. So, disaccharides are 2 monosaccharides joined together by a covalent bond. A covalent bond is just them sharing these things called electrons, which are negatively charged particles in their electron shell. The most common disaccharide is going to be sucrose, as we see depicted down at the bottom. It is the monomers of sucrose are going to be a glucose and a fructose. Those come together and join to form that sucrose molecule. Plants also transfer carbohydrates from their roots to their leaves in the form of sucrose. Now we're going to talk about polysaccharides, and polysaccharides are a polymer with many sugars joined via a dehydration reaction. So a polymer is many sugars, and they're joined together by the removing of water. We have 2 different types of polysaccharides. The first one we're going to talk about is storage polysaccharides. Plants store starch in the form plants store carbohydrates in the form of starch. Starch is just a polymer of glucose monomers. So it's just a bunch of glucose bonded together and stored. This allows plants to store that extra glucose that can be used later on as food for their body. Then animals store it as glycogen. Wow, English struggles. Which is, once again, just a polymer of glucose, so many glucose sugars put together. And these are stored in the liver and muscle cells of animals, including you. We have structural polysaccharides, which we can see in plants as cellulose, which is a tough substance that forms that plant cell wall that helps keep that plant cell from shrinking or expanding too much. And we can also see it as chitin, which forms the exoskeleton of arthropods, or that hard outside shell. That crunch you hear when you step on a cockroach is that chitinous shell. So, here's your first question. I'm going to give you a second to answer it. I knew you guys were going to get it. So, sorry, I went too far. So we are going to move on to proteins. Proteins, the formation of a protein happens in a couple of steps. We've got amino acids, which are small individual protein, not even a protein yet. Then they come together and form a peptide. Then those peptides join with other peptides to form a polypeptide. And those polypeptides come together and fold within each other to make a protein. Proteins are super important. They're what we use to live and perform most of our function every day. So a protein is just a molecule that contains a bunch of polypeptides, which are polymers of amino acid that are folded into a 3D shape. These guys are comprised of carbon, hydrogen, oxygen, nitrogen, and sulfur. The shape of the protein determines the function. So structure dictates function. Let me repeat that again because it's super important. You can answer most questions just knowing this fact. That structure dictates function. So, what is an amino acid? We've got 20 different ones. So they're a molecule with an amino group and a carboxyl group. There are 20 different amino acids, and they all have roughly the same general structure. So you can see here we've got an amino group on one side that has that nitrogen, and that carboxyl group on the other side that has that oxygen and the oxygen and hydrogen with that carbon. And then we have an R group, which is just a variable side chain that can be changed depending on which amino acid it is. So we have two examples. We have alanine, which you can see, once again, we've got that amine group here, carboxyl group here, and that R group is now a CH3. But if we go over to glycerin, we have our amino group here, our carboxyl group here, and just an H here in that R group. So that R group is always going to change and be something different. You see that they're unique. Each amino acid has that unique side chain. The unique aspect of the amino acids are based on that side chain's physical and chemical properties. And side chains can be grouped as nonpolar or hydrophilic, meaning that they share charges equally and they really hate water. Polar, so they don't share charges equally, and hydrophilic, meaning they really like water. And charged or ionic and hydrophilic, meaning once again, they really like water and they have a charge. So they're either positive or negative. And that side chain determines the interaction, which determines the shape and then, therefore, the function of that protein. So, what happens when two amino acids come together? So they form a polypeptide, a peptide bond. To form a peptide bond, the carboxyl group of one amino acid must be positioned next to the amino group of another amino acid. And then dehydration happens. And dehydration is the removing of water, and they become friends and they're bonded together and they make a peptide bond. So, polypeptides are many, many, many amino acids linked together by peptide bonds over and over and over again. Each polypeptide has a unique amino acid sequence and is directional. So you can only read it one way, like we read books left to right. Each end is chemically unique. One end is a free amino group, and one end is a free carboxyl group. These respectively are called N terminals and C terminals. The sequence of amino acids, so how they are strung together, determines that 3D shape. Remember, structure dictates function, or shape determines function. Whenever a polypeptide twists and folds because of that R group interaction, it forms a protein. And we have different names for each one of those twists and folds. Quick. Now, I want you to think, pair, share, and write your answer in the box about how is a unique sequence of amino acids determined for a peptide bond. That's right, genes. So, why are proteins so special? What are their functions? The functions of proteins include antibodies, so they help protect the body from diseases. Enzymes, they carry out chemical reactions or assist in creating a new molecule. Messenger, so they transmit signals, i.e., hormones, throughout the body. Structural, they provide structural support for our bodies. So they create some parts of the cell wall. They help control what comes in and out as well. Transport and storage, like I just said, they bind and carry small atoms and molecules throughout the body, in and out of cells. So there are levels of protein structure, and this is the cool part. So we have our primary level, which is just a straight linear chain of amino acids, like you see right here in this photo. They're determined via genes. So genes, it's called the central dogma. DNA makes RNA, which makes proteins. And we'll talk about this a little bit more later on in biology, but I just want to put that idea out there for you. This dictates, this linear chain of amino acids dictate the secondary and tertiary forms of that protein because we like those side chain interactions. So, secondary are coils and folds due to hydrogen bonding. And hydrogen bonding is just water molecules interacting in a special way that makes things kind of stick together and are really interesting to pull apart with a polypeptide backbone. So they've got a backbone, like a spine of polypeptides, and they're folded in on each other with those hydrogen bonds or that water bonding. So we have beta pleated sheets that have hydrogen bonds between the polypeptide chains lying side to side. And we have alpha helix sheets, which are hydrogen bonds between every fourth amino acid. So we can see here it almost looks like a wave or pleats in an outfit. And then our alpha helix is like a corkscrew, twisting in and of itself. So beta pleats, alpha helix spinning. It's the fourth, and then it's between them laying side by side. So then we have our tertiary structure. And this is where the 3D folding happens due to the interactions between the side chains of the amino acids. They are reinforced by that hydrophilic interactions and disulfide bridges. So it's a 2-sulfur bond of those side chains, and they don't like water, so they're going to try to skirt and not be around that hydrogen bonding. And this is where a covalent bond, where they share electrons, are formed between sulfur atoms of 2 cysteine or secondary monomers. And then we have our quaternary structures, which is the fourth level of folding, which 2 or more polypeptides come together and intermingle. They are found only in some proteins, so we're not always going to see quaternary structures. All 4 levels of protein structure determines that protein's function. So some are only going to be seen at tertiary, some are going to be at quaternary. So we can see in tertiary, we've got some beta pleats and some alpha helixes to help determine that shape. It's just where they line up and how those bonds interact. So we're going to pause here and pick up here on Monday.